Today we'll be explaining DNA replication in prokaryotes and the function of specific enzymes during this process. DNA replication occurs in the S phase of interphase.
There are 5 main enzymes essential to the DNA replication process: DNA helicase, DNA polymerase III, RNA primase, DNA polymerase I, and DNA ligase.
First, DNA helicase unwinds and unzips DNA. The helicase breaks the hydrogen bonds between the base pairs, leaving them exposed. Whilst DNA helicase unwinds the DNA, DNA gyrase relieves the strain. DNA gyrase is present in prokaryotes and is a type of topoisomerase, whereas helicase is not.
The unwinding of DNA occurs at specific regions, which creates a replication fork of 2 polynucleotide strands in antiparallel directions.
RNA primase then follows the helicase and leaves short RNA primers on each parent strand to provide an initiation point for DNA polymerase III on the lagging strand.
Then, DNA polymerase III adds deoxynucleoside triphosphate to the 3' end of the chain, which allows the process to occur in a 5' to 3' direction.
The deoxynucleotide triphosphates pair up with their complementary base partner, but as they join, 2 phosphates are broken off, which releases the energy needed to form a phosphodiester bond.
The complementary base pairing rule is that adenine always pairs with thymine and guanine always pairs up with cytosine. This ensures that the base sequence of nucleotides is conserved. DNA polymerase moves in the opposite direction on each parent strand.
Meselson and Stahl proved that DNA replication is semi-conservative. This means that new strands contain one original parental strand and one copied complementary strand. As stated above, complementary base pairing ensures that the base sequence of nucleotides is conserved.
The synthesis continues on the strand moving towards the replication fork, which lies on the leading strand. However, it discontinues on the strand moving away from the replication, located on the lagging strand, which leads to the formation of the Okazaki fragments.
The Okazaki fragments are sections of copied DNA which lay between RNA primers. When it reaches another primer, it detaches and leapfrogs to the next primer.
DNA polymerase I moves along replication fork, moving the RNA primers and replacing them with DNA.
Finally, DNA ligase attaches to the Okazaki fragments into a continuous strand of DNA, and DNA is fully replicated.